Hello and welcome to Chapter 4 in Biopsychology, where we are going to be looking at the anatomy of the nervous system. This is the brain, spinal cord, and the rest of the nervous system, kind of divided into the central nervous system, CNS, and the peripheral nervous system, PNS. So here we are at Chapter 4, and we start off with a discussion of the central nervous system. And we're going to actually start off here with an exploration of the spinal cord. And what I would like to do is spend a little bit of time talking about the anatomy of the actual cross-section of the spinal cord, pointing out the structures that are there. These will be relevant later in the class. And then take some time looking at the designation of C for cervical, T for thoracic, and for L for lumbar, and S for sacrum as designations along the spine, and why these have relevance in the world of psychology and healthcare. Just some storytelling in that degree. So let's take a look here at the anatomy. Here's a cross-section of a person's spine, and we start off, we have the actual body of the vertebrae, the bony actual structure. This is where the discs are. This is the structural bone that makes up the majority of the spinal column. We can see that it's made up of bone vertebrae here, and we have inserted in there, we have these nerves, these spinal nerves that come out in spaces all along. They actually branch out from the spinal cord as part of the peripheral nervous system. And when we hear of somebody having a pinched nerve in their back, that means that there's been some inflammation or there's been some damage to the actual bone structure in there, and these nerves are being pinched. And you can obviously, these nerves are major sets of neurons coming out, going to parts of the body, and not only would you have pain in the localized area, but it would interfere with the functioning to the part of the body where those nerves are going to. Then we have up here, we have the actual spinal cord itself, coming from the brain all the way down to the tail, made up of white matter. Now remember, that is myelinated in order to increase the speed and efficiency of those signals, and gray matter, which is not myelinated. So it's a combination, and just like in the brain, we have the myelinated fibers inside, primarily made up of the corpus callosum, which we'll get into in a minute. And then on the external part of the brain surface, we have unmyelinated nerves, the gray matter. And then we have some structures over here. These are protective tissues that wrap themselves around the spinal cord, and they're very similar to the meningeal layers that surround the brain. And the pia mater is actually a very delicate membrane just wrapping right close to the actual spinal cord itself. The arachnoid is a kind of spider web. That's why it has that name, arachnoid, a spider web structure that's actually like a shock absorbing suspension system that allows the spinal cord some movement without damaging. And then a very tough dura mater, which is a membrane on the outside of that suspension system. And we see this kind of nomenclature here. We have arachnoid, this from arachnid, which is the word for spider, the spider webby kind of cushioning, and dura matter, like as in durable. And so, having spoken to a couple individuals that have been involved in some surgeries, either spinal cord surgeries or brain surgeries, that this dura matter is actually really tough. Like even with a scalpel, you have to kind of put some pressure to get through that. It's a very, very tough fibrous material that's on the outside of our brain, but also down into the spinal column. So let's take a look at the column itself here. We have sort of this curvy bone structure here, and it's designated by sections that have to do or correspond with function in the body. So you'll see way up in the top, when you think about those nerve fibers that are coming out of the spinal cord, you know, right from the top, there's nerves that come out, and there's nerves that come out all the way down. Where do those nerves go? And of course, they're logically located, so as you go down the spine, those nerves go to lower and lower parts of the body. That's where the signals go to control those muscles or to control those parts of the system. So the ones that are up here are going to control the top body. The ones down in my tail are going to basically control my legs. And so they operate in a very logical sequence. So we can see here that we have head and neck up here, head and neck, and then the diaphragm very, very up there. Now that's an interesting one right there. The diaphragm is actually the breathing mechanism way down in the next to in the abdomen. So a little bit different in terms of the sequence there. And we have the biceps and the wrist and triceps and the hand, all in the cervical nerve areas. And then we look down into the thoracic area here. We have the chest muscles, the abdominal muscles, and moving even further down, we have lumbar leg muscles in the lumbar part of the spine here. This is why if you don't have lumbar support, let's say on your chair, in your office chair, or lumbar support on your car seats, that sometimes your legs fall asleep. And so that those nerves get, you know, pressed upon, and there's pressure on them, and then they begin to tingle, and they start to not operate well, and then we lose some feeling in our legs, quote unquote fall asleep. Now it's also interesting to look at this particular diagram and consider that we might refer to any one of these, let me go back here, might refer to any one of these here as we may hear of someone who's had a spinal cord injury, and they'll say it was a C8 spinal cord injury, and that what that means, very logically, is that the person had a damage at this level of the spine. Or they may have had, let's say, a T8. And we could then diagnose what functional aspects might they be having trouble with. So an individual, let's say, who has trouble way up here is going to have this level, you know, when you have as C1 through C3 type injury, we're really talking about right at the base of the skull. Many individuals do not survive that. If they do, those individuals they lose mobility in their entire body, they lose feeling in their entire body. They'd be considered a quadriplegic, an individual who has 4 limbs, all 4 limbs, are as opposed to a paraplegic, which is only 2 limbs. Let's say they get a thoracic injury or a lumbar injury, they would lose function of their legs, but they would still have motion in their arms. Now, as adaptive as the nervous system is, it's not a cut and dry sort of prediction, but there will be functional deficits according to the degree of injury. So we often refer to a person's spinal cord injury by referring to the letter corresponding to the vertebrae where the damage occurred, and thus we can say, you know, someone is a T5, or someone has a T5 spinal cord injury or something like that. So useful terminology to understand if you're ever having to deal with that. Now the next part of the central nervous system, moving right along here, of course, is the brain itself. So the central nervous system is made up of the brain and spinal cord. We're going to be talking about the peripheral nervous system next. But let's take a look at these major brain areas and how we understand their particular functions. So we start off here at the cerebral cortex, that very familiar outside covering of the brain, the gray matter. Ultimate control for information processing, executive functioning, processing some motor movements, processing some visual stuff we'll see in other parts of the cerebral cortex. When we look at the occipital lobe, we will be looking at parts of the brain throughout this course and how they act, right from the get-go, we have this sort of myth out there that we're only using 10% of our brain, but in all actuality, we use our entire brain, and in fact, it's amazing how much of our brain we use for each one of the things that we do. So like when we get into how we process visual information, we're going to see the whole brain gets involved, not just the occipital lobe. You see that messages go to the entire brain, and it works as a unit to create, what we have as a visual experience. So we have the cerebral cortex, again, the ultimate control and information processing center. We move down here to these areas here. We have the hypothalamus. It's inside it's kind of almost above your palate in your mouth. Good. Above the soft palate, that's where the hypothalamus is. Controls maintenance functions like eating. They govern the endocrine system, linked to emotion and rewards. So this is emotional food and emotion and hunger and satiation and those kinds of things related to the hypothalamus. We have the pituitary, one of those glands that is actually secreting hormones like we talked about in the last class. The midbrain, this is very, very basic functions of the brain, motor movement of the eyes, audio, visual processing at a very, very basic level. The information comes in. If you think about the pathway, as we will talk about when we talk about the auditory pathway, talk about the visual pathway, they all go to that central part of the brain where then they branch out and send information to the sensory areas for processing in the cortex. So we'll be examining those, but here's that midbrain area. And we have the pons. These are involved in the control of breathing, sensations such as hearing, taste, and balance. You see, the pons sit right up on top of the brain stem, which is the connective tissue to the spinal cord. And we shift over here. Oh, I put my little arrows here. I forgot I did that. And there's the pons. I'll remember it this time. So here's the spinal cord. This is the pathway to the neural fibers here. And then that connection that is the bottom part of the brainstem here, the medulla oblongata. Now this helps regulate breathing, heart, and blood vessel function. This would be, if you had a very, very high spinal cord, like a right up at the top, it could affect the medulla oblongata, and there's almost no ability to survive that. It's that's a very, very high level injury. And so damage to this area, look what it controls. It controls your heart, breathing, whatnot. We have the cerebellum, coordinating voluntary movements, balance, and the memories associated with this. So this is your riding the bike memory is kind of in the cerebellum, the actual physical memory or muscle memory that individuals have. So athletes, or when you learn how to type, whenever you learn something that's automatic, you learn how to drive your car, and everything becomes automatic. It's because the memories of these motor functions are stored actually in the cerebellum. So we locate those there. We're back into the inside, what we call the basic inside structures of the brain that are shared among all mammals, the mammalian brain, and we have the amygdala linked to emotions. And we're going to be talking a little bit, we talk about it in developmental psych, and we talk about the amygdala as an emotion, pleasure-seeking part of the brain, kind of really basic, selfish part of the brain. And then we have the prefrontal cortex, which is the aspect of the brain that makes for logical decision-making and prioritizing and planning and executive functioning and all that. And we see that there's a developmental process, whereas when you were born, those pieces are not talking to each other, basically operating independently. And as we age, and sort of peaks around adolescence, somewhere around adolescence, the connection is made. But leading up to that point, it's difficult for an individual to not simply seek out the sort of what Freud would call the id impulses that are for pleasure seeking and selfish motives and stuff like that. So we see this egocentrism associated that we see outside, just sort of like, why is everything about you? And it really is because that prefrontal cortex is not connected to the amygdala, so there's no reins on the emotional needs of that individual, and so they can act in highly selfish ways. And we see that biologically, interesting aspect to that. And this is the part of the brain, the amygdala, that we want to rein in. And so that's something that we look at in developmental psych. And we move up here, we have other parts of the brain, the hippocampus related to memory. This is kind of a circular structure around the amygdala. We have the fornix, acts as a major output tract to the hippocampus from the other parts of the brain. The thalamus, relaying messages between the lower brain areas and the upper brain areas. So you can imagine how important that is. This is the connective tissue that eventually, depending on where it's growing, it's the thalamus that will send out messages to that prefrontal cortex and connect them with the amygdala. And then finally we have, as I mentioned a little bit earlier, the corpus callosum. Now this is axon fibers connecting the two hemispheres, largely made up of highly myelinated nerves. Unlike most of the rest of the brain, which is the gray matter, we have the corpus callosum, highly myelinated. We need that speed, in order to process information from one side of the brain to the other. And this is a fibrous tissue that connects the two different hemispheres. So how did we all discover this? Now, this is something it's a very interesting part of the history of psychology class, where we go into how did we figure out the functions of the brain. I mean, all those labels are on the picture, but then when you open up the brain, there's no labels. And so how did we make these connections? And throughout history, there's been different methodologies that we've used to do that. And they include post-mortem studies, and that means we've been able to observe people, and then there are opportunities when we have the ability to then examine their brains after they've died. And then we kind of make a connection. Now these individuals in the past, there was lots of issues associated with looking at bodies of people that had passed away. In fact, in the kind of the early science of this, the only ones that we were able to examine were convicts, prisoners that had died. It was considered under the church sacrilegious to operate on a dead body. It was considered sacred, and so we'll just do it to the people that are going to hell already. So this is really kind of the thinking that was going on. So criminal bodies and whatnot. And so kind of the first instances of this kind of exploration of the functions of the brain was trying to figure out criminal behavior. Why were these individuals acting out? Why were they doing that? Trying to make correlations to the brain. Of course, things like blindness and lack of hearing could be correlated with parts of the brain that were damaged or something like that, or somebody that had a brain injury and we can see their behavior was different. Then we look at the brain and we're like, oh, there's the part of the brain that was damaged. That must be why they were acting this way. And over the years, we sort of map out portions of the brain this way. Now, the other way in which we have done this has been extirpation. And what this is, mostly with animal studies, I'm going to say mostly because I suspect that it's probably been done to some humans as well, is actually the purposeful lesion of parts of the brain. Now, in animal studies have been done where they've operated on the brain and they've destroyed a piece of the brain, and then they watch to see what happened. Now, this seems and is a pretty cruel way to learn about the brain, but nonetheless, that's part of the history of psychology, is this. And it's not necessarily ancient either, as we'll see in the next example here. And another way that we examine functions of the brain is actually to measure or introduce electrical stimulation. And we know that as we know from the neuron, it operates on an electrical principle. So if we put a small electrical charge and an individual acts in a certain way, an animal or a human being or whatever. In fact, when there are occasions when individuals are undergoing brain surgery that the patient is kept alive. Of course they're kept alive. They're kept awake. And the doctor can stimulate parts of the brain in order to locate precisely where they need to make their incision based on the experiences that people have while their brain is being stimulated. So they may stimulate a part of the sensory cortex, and the individual will report, I'm smelling roses. And the doctor then knows, well, I'm in the sensory cortex. Certainly advanced medicines and brain imaging now has reduced the need to do that sort of, walking through the woods to find something methodology. But nonetheless, we've had that. Now, I say this is an ancient history, although when I went to college, it was a long time ago. In 83, when I went to school, I ended up taking a physiological psychology class, and part of that class entailed using a rat lab to conduct neurological experiments. Now, the truth is, I engaged in experiments. I had the Red Baron, my rat. We were all given a rat. We had to take care of the rat and feed him and clean his cage and all that stuff. And I named him the Red Baron because he had red eyes and pure white, big old white rat. Very nice animal. Wasn't gross or anything like that. And we had to conduct a study where we would have the rat go into it. It's a mechanism that holds their head. This is, using rats has been in part of psychology for decades. So there's a mechanism that holds their head in a particular way, and we use a angled arm to determine exactly where we're going to insert a electrode to measure brain waves. Now, I chose an experiment working on the occipital lobe, and I was going to run an experiment, a classic maze experiment, to record activity in the rat's occipital lobe while it was negotiating a decision-making maze. Basically, the rat came to a divide in a very simple maze, shaped almost like the goalposts on a football field, going straight. On one side there was one color, on the other side there's another color, and the right color was a one-way door. They would go through that one door, and they would either go to food or no food, and training the rat to recognize which color meant there was food, to associate that. And so I did that, and we measured the electrical activity. However, because this is very, very small areas of the brain, very, very delicate and very precise areas of the brain are being measured by these tiny little wires that are inserted into the rat's brain, we don't actually know which neurons we're actually measuring. So in the end, what we had to do was reverse the polarity of the inserted electrode and burn a lesion in the rat's brain that would mark the end point of the electrode. We then would kill the rat, we would remove its brain, we would freeze it, and then we would slice it in small slices until we can find that little lesion. It seems almost archaic, doesn't it? But I did it, and I won't go into all the details about all of that, but we did find that I had a lesion in the occipital lobe, and the measures that I had in my test was successful in terms of measuring increased activity in the occipital lobe when my rat was contemplating, do I go left or right? And so that kind of process of both measuring and stimulating, and sometimes that extirpation of destroying a part of the brain, in this case, not to see how the rat would act, but to mark the location that I was measuring. So clearly, some ethical issues in terms of the treatment of animals. So moving right along from my past as a rat killer, let's talk about the peripheral nervous system. Now, this is essentially everything else. All the other nerves of the body, all that extend, everything that's not the brain and everything that's not the spinal cord, even if the nerve is really close to the brain. Like the nerves that go from our ears to our brain don't have to travel very long, but those nerves are part of the peripheral nervous system. Everything that's outside the brain is that, just like it's as peripheral as the nerves at the end of your big toe. And so those are all part of the peripheral nervous system. Now, the peripheral nervous system is divided into sections. Psychologists love to do this. We have the somatic nervous system. This is the system of nerves, the call and response, I guess, the purposeful motor movements, the body movements that we make willfully. It's also part of the sensory system. Here we have, we can move, I can make my arm move, so that nervous system that allows me to move my arm, allows me to vocalize, allows me to turn my head and move my eyes, that's all part of the somatic nervous system. Next to that, we have the autonomic nervous system, which takes care of all the automated things that, thank God, we don't have to worry about. We don't have to think about breathing. We don't have to think about digesting. I can't imagine in a world where we'd have to, okay, I got to breathe now. I'm feeling lightheaded, kind of we breathe all on our own. We digest food. Imagine a world where we had to think about that. We'd have dinner, and then we'd all go into this like cubicle and just like, okay, I'm digesting now. We don't do that because it does it for us. All of these things that happen. Now, in that autonomic nervous system that is managing all of our automatic functions, there are two systems at play. One is an activating system that responds to stimulus from the outside that says, I need to beef things up, various functions. And one is a sort of a relaxing or suppressing system. And we refer to those as the parasympathetic system and the sympathetic nervous system. Now, the graphic I have for this one, kind of a complex one here. Here we have the parasympathetic nervous system on the left. We have the sympathetic nervous system on the right, and we can add some labels here. The parasympathetic is inhibitory. It's toward relaxation of these mechanisms. And then we have the sympathetic nervous system, which is excitatory, responding fight or flight. Now, what happens is when our nervous system is excited and it's stimulated, someone cuts us off, let's say, it activates our sympathetic nervous system. Our pupils dilate, we get dry mouth, our heart rate increases, it inhibits digestive activity. We might feel like we have a rock in our stomach. Stimulates glucose release in your liver, so you're getting some little bit of a high off of that with the sugar high. Increase in epinephrine and norepinephrine releases. Could relax the bladder. You could have an accident, and same of course with lower parts of the body and reactions to that. You see these excitatory aspects of the nervous system here that would respond to a sudden threat or a sudden. But now again, you got keeping in mind that excitatory processes happen not just because we're being a threat, I mean, we can have excitatory processes because of sexual activity, excitatory processes because we just ate, and that's going to be something that we have to react to. But nonetheless, that's sort of the fight or flight one. Now then as soon as we've passed through this sort of phase here, we go, the parasympathetic system kicks in and starts to relax the body. And we see the pupil constricts. We start to salivate. Our heart rate decreases. Our body can then start digesting food. Gallbladder is stimulated. The production of adrenaline is inhibited. Contracts our bladder. Our rectum is relaxed. This is the body calming down. This process. I mean, when I've dealt, the way that I've encountered this has been, of course, in crisis management, when an individual is in a psychological crisis and their excitatory fight or flight system has been activated. The fact is that I might be able to have them make some better decisions, in terms of the crisis intervention, but the biology sticks around for a while. And you have to, when you're doing crisis intervention, really recognize that even though the individual may appear calm, their body's excitatory chemicals are alive and well in their body for up to 24 hours after that. This excitatory sympathetic nervous system injects the body with chemicals that will affect it for up to 24 hours. The lesson in that is that when you're dealing with someone who's in crisis, even though their behavior may have calmed down or whatever, it won't take much to get that person back up again. They're in sort of a danger zone or a risk zone of becoming worked up again, and you have to be careful of that, in terms of managing what that person is going to be doing for the next, or helping them to manage what they're going to be doing for the next 24 hours or so, while their parasympathetic system is kicking in. We activate our parasympathetic system. So let's say somebody cuts us off and we're all worked up, and we pull over and we start doing some deep breathing and focusing. I might close my eyes, I might rest my hands. I'm focusing on actually purposefully engaging the parasympathetic system in order to calm myself down. So that's the process there. Now let's take a look now at the assessments that are due for this particular chapter. We have a discussion on animal studies and why we still use animals and should we, and having a discussion about that. We have a quiz really just asking, what are the advantages of white matter in the spinal cord? So coming up with an answer for that. And finally, we have an assignment. And this is kind of open-ended. And what it entails is creating a slideshow with a verbal narration, recording a narration on top of it. So you create a slideshow. You can create it in PowerPoint. If you have a PC, you can create it in Keynote. If you have a Mac, export it as a movie and upload that. It's a different way of presenting. And what you're going to be doing is using your own graphics, using your own research, is teach a lesson on the parts of the brain. Go into as much detail as you want. Has to be pretty sufficient detail, a little bit more than I did actually. Go into some of the functions of the brain. And the idea of this kind of activity is that if you practice teaching, as I do, as I teach this is a new class for me. I get a little bit better each time. And if you have the opportunity to teach somebody the parts of the brain, it helps in terms of remembering those things. So I'd like you to do a presentation on brain parts, functions, the anatomy of the brain, not the spinal cord, not the peripheral nervous system, just the brain. So that's the assignment that goes along with here to help you remember parts of the brain like this. So that's it for this chapter, and have a great week.